When we do trials, whether they're on farm or on station, we get our data sets. And before we make a recommendation to farmers, we do two things. We do a statistical analysis and we do an economic analysis to see if it's profitable for farmers. And today I'm talking about the statistical analysis. And the reason we do the statistical analysis is that we want to know how confident we are that the observed yield differences between two or more practices were not due to chance. And we use statistical analysis to determine our level of confidence. And these level of confidences that we work with depend upon our purpose. If we're doing it for scientific purposes, we want a high level of confidence that we're right when we make a conclusion. If we're making decisions at the farm level, we can be satisfied with the lower level of confidence. These data are from a trial conducted on farm in 2012 to look at the effect of plant population on corn yields. And so we have 30,000, 34,000, etc. And what we see is there's a lot of variation in the yield, that each strip or plot has a different yield. So how do we deal with this variation? We want to know how much of that variation is due to the plant population effects. And if we see here, we see that there is some difference due to plant population, with a relatively higher observed yield with 34,000 plants than with the other plant populations. But some of the variation may be due to variation in the field. So if we compare the yields of Rep 3 to the yields of Rep 4, we see the yields of Rep 4 on average are higher. So we do have some variation in the field. And then we have a lot of unexplained variation where we can't account for it, and we call this experimental error. To sort this out, we use a procedure called the analysis of variance, and it helps us to determine how much of the variation is due to plant effects, plant population effects, how much is due to variation in the field, and how much is unaccounted for, what we call experimental error. And with this, we can determine what is the probability that the observed plant population effects are repeatable under similar conditions. So we've taken this data set, we put it in the statistics program, and we have different statistics programs available for us for this analysis. This is where we've taken the variables and assigned them to different categories. So we have our dependent variable, which is yield, and to some extent it's dependent on our rep and our population. And then of course there's the experimental error. The results look something like this, where on the left-hand side over here we have our sources of variation. How much is coming from the replications or the blocks? How much is coming from the plant population? How much is due to experimental error? And then for each of these we get a variance term. So it's 23 for the error, 14 for population, and some for the reps. Over here we get this number P, which really tells us how much confidence we have in our results. If here we have 0.6, we would say with this, our observed differences are not statistically different. We see down here that there is some difference, but there's too much variation in yield from plot to plot to be able to say that they're statistically significant. So these were the results of one trial. To have good confidence in our data, we need to repeat this work over multiple sites or multiple years. And this trial was conducted at 3 locations last year, same treatment, same number of reps, and we get a more complex ANOVA output in this case. And again, here we have a large variance term associated with locations, so it tells us that the different fields had different yields on average. But our population effect here is only 7 compared to an error term of 70. And if we look at the probability of differences, we see that it's going to be very small. If we subtract this number from 1, it tells us that we only have about a 4% chance of getting similar effects if we repeat this research under similar conditions. So here we do see the means from this. We have the 3 locations. We see a relatively low mean for C1 compared to H2. And we have our population means, where the yields are either 26 or 27. So very little difference due to plant population. And again, we conclude that these are not statistically different. So in summary, we analyze the variation in trial data to determine our level of confidence, that is the probability that observed differences between treatments are due to treatments rather than to chance.